Hello, everyone. For this AIRC seminar, this is the third time this year that we are meeting, and we will have two very exciting speakers. We have Frederick Matsen, who will talk about inviting Darwin into antibody foundation models. And then afterwards, as the early career speaker, we have Lena Erlach from ETH Zurich, and she will talk about antibody affinity engineering using machine learning. To briefly introduce Eric, I think everybody knows Eric in the air field, but just very briefly, Eric trained as a mathematician and then evolutionary biologist, and now works on adaptive immunity, mostly developing probabilistic models of immune repertoires. He's a professor at the Fred Hutch Cancer Center and also a Howard Hughes investigator. And thanks so much, Eric, for being here. And I'm looking forward to your talk.
Great. Okay, I'll share my screen. That look all right?
Yes, looks great.
Awesome. Well, so there's a frequent theme in artificial intelligence research that people say that all you need is a really big model and a lot of compute to solve any problem. And the pitch is tempting. Don't worry about the underlying biology. With enough data and enough GPUs, the model will figure it all out. Today, though, I'm here to argue the opposite. When we incorporate fundamental biology into machine learning models, we get predictions that are much more accurate, and the models are smaller and faster and more interpretable. So I'll be talking about antibodies. These are amazing molecules that can bind just about anything to high affinity and specificity. As people here well know, antibodies are created by B cells. And when they're displayed on the outside of a B cell, they're called B cell receptors or BCRs. They emerge via random VDJ recombination of genomic DNA. And the goal of these B cell receptors is to bind antigen, which I've schematized as this red circle. And an antigen is just a general name for something that an antibody wants to be able to bind. It could be part of a vaccine, or it could be part of a virus, or anything else. So after the B cell has bound antigen, and I'm sweeping a lot of things under the rug here, as everyone here is aware, it stimulates a process of mutation. And sometimes these mutations, like this yellow mutation, will improve the binding of the antibody to the antigen. And sometimes, such as the silver mutation, it will remove the binding to the antigen. After a mutation, there's also a selection process in which the B cells that can bind antigen are allowed to reproduce, and those that do not bind antigen can die. And this is repeated many times. So one of the things I love about working in this field is that we can think about antibodies from two perspectives. The first perspective is that of the natural immune response. We can take a blood draw from someone and see all the things they've been exposed to implicitly by sequencing their collection of B cells that encode the antibodies that they're currently making. So these are the memory B cells. So we get a big stack of sequences. And now there's over 1 billion sequences that are available in public data sets. And this is in part thank you to we can have that natural perspective on antibodies, but we can also think about antibodies as being protein molecules with functional properties such as stability and binding. And one might think if you have this huge stack of BCR sequences, surely that's going to tell us something about this other situation, about the antibodies as protein molecules. And indeed, the community thinks so, and they think that we can use cutting-edge machine learning frameworks to do this transition from the natural side to the functional prediction side. So here is a list of some of the current antibody language models. The modern ones have hundreds of millions of parameters, or a billion parameters and are trained on hundreds of millions or a billion sequences. These language models are trained as follows. Let's take a naturally occurring amino acid antibody sequence. We mask out an individual site, and the model is asked to predict what that missing site was. So it predicts it probabilistically by assigning a probability or a likelihood to each one of the possible amino acids. The objective of this model training is to maximize the likelihood of the observed amino acid, which in this case was Q, across all the different possible maskings and different sequences. So after training this with this sort of masked language modeling thing, these models can get evaluated in papers such as this. So I'll call this the FLAB paper. And in order to describe the types of benchmarks that people do, I need to explain one mathematical term, which is called perplexity. So perplexity is actually a great term for this because perplexity is really a measure of how surprising is the sequence according to the model. How perplexed does the model get when it sees a given sequence? So the equation looks like this. If it looks a little overwhelming, don't worry. So first, this outer part is just a type of mean. So rather than an arithmetic mean where you have a sum and a 1 over n, we have a product and the nth root, but it's just a mean. So what are we taking the mean of? We're taking the mean of 1 over this probability. And this probability is, what is the probability of the observed amino acid given the rest of the sequence? So we're taking the product of over all sites of, again, 1 over the probability of the observed amino acid at that site given the rest. So just to break it down a little bit, if the model is absolutely sure that the observed amino acid is the only possible amino acid at that site, then it's going to give that probability, that's going to be given a probability of 1. So 1 over 1 is 1. So that's a perplexity of 1. And that is the absolute minimum perplexity we can have. You can imagine a situation where the model is guessing randomly. So we're going to have a probability of 1 over 20 of each of these amino acids. And so 1 over 20 is just 20. So that sort of gives you two different basic baselines of like, 1 is the best possible you can get, 20 is random guessing. And if you get larger than 20, that means the model is assigning a low probability to the observed thing, lower than just random guessing. And if this sounds a lot like masked language modeling, it totally is. You can think of masked language modeling as really just minimizing the perplexity. So with that background, we can now look at one of the figures from the FLAB paper. This is the type of evaluation that they're interested in doing. On this axis is the melting temperature. So this is some biophysical property. And generally, I'm just going to, when I say function in this talk, I'm going to say some biophysical property such as stability or binding. So I'm just going to use that in a pretty general sense. So this is our functional property. And on this axis is the perplexity. So the surprising sequences down here, or sorry, the not surprising sequences are down here in the lower part. So these are the ones the model expects or make sense. And then the ones up here are the ones that the model thinks are more surprising. Over here are the stable antibodies, and over here the less stable antibodies. So this is viewed as a success because the ones that the model thinks are compelling that are not surprising are also the stable models, stable antibodies. The ones that are over here are the ones that are unstable and also surprising according to the model. So here, the model is doing a good job of telling us something about the biophysical properties of these antibodies. So this sort of makes sense. So we can imagine that the models are telling us something. They're basically modeling the empirical distribution of all the antibodies that are out there in nature. And the ones that are in nature tend to be stable. And so it sort of makes sense that you would be able to use this as a predictor of stability. But there are some flies in the ointment. And I really credit the Oxpig team and Charlotte Dean's team for being the first ones to raise, I think, an interesting question. And if you haven't seen Charlotte Dean's Air Talk, it's amazing. Please go back and watch it. So when they published their Ablang 2 paper, they raised a question: should we really expect masked language probability to correlate with function? And they showed a table. Oh, yes. And the thing that they were remembering is that amino acid sequences of antibodies don't come from thin air. They come from a specific process that we know things about. And it particularly comes from random VDJ recombination of genomic DNA. And when you have this random genomic recombination, there are some sites that come directly from your parental genome. So some of these sites will come directly from your parents, from your mother or your father's germline DNA. So they made a table in which first they stratified the perplexity calculations for these different language models according to whether they were germline residues or not germline residues. So I want to be clear what germline residue means in this context. It is a amino acid at a given site that comes all the way from genomic unrearranged DNA. So a certain amino acid was not mutated. It was not involved in VDG recombination. It came straight from your mother or your father. So that's what they consider to be a germline residue. And you can see that these perplexities are amazingly low. Here's a perplexity of 1.01. That's crazy. But you have to think about, what is this model actually doing? So what it's doing, all it has to do to get a good prediction is to look at this partially masked sequence, match it up with what it sort of has learned are the germline genes, and restore the germline amino acid from that table. So although it doesn't directly have a lookup table with its millions of parameters, these models have no trouble memorizing what the germline genes are and restoring them. So that's why you get this outstandingly low perplexity. On the other hand, if you look at the non-germline residues that appear via VDJ recombination or mutation, the numbers get from incredibly small to embarrassingly big. So remember, 20 is just random guessing. So these pretty sophisticated models are getting worse than random guessing on the non-germline residues. So what they did, Charlotte Dean's group did, is they changed their loss function to something called a focal loss, where they're focusing more of the loss on the non-germline residues. And you can see that had an immediate and improved response. So now the perplexities go down to about 10-ish. So that's great. And I really want to credit them for raising these questions and coming up with a solution. We're going to be looking more at Ablang 2 now and poking at it a little bit. And it's not because I want to make fun of Ablang 2. It's representative of these models. And it's the best model out there because it does incorporate this type of biology. I just want to point that out because we're going to be poking at some weaknesses of Ablang 2, but I'm going to be saying Ablang 2 a lot, but it's not a weakness of Ablang 2. It's a weakness of the masked language modeling criterion. So I think that Ablang 2 is really a substantial advance. And the thing that these models actually, I think, sort of forget in general is that B cell receptors are coded for by DNA. And we know things about DNA. We know about the codon table. We know about transcription. We know about all these things, and we know about mutation. And what I'm going to show is that these masked language models have implicitly learned things about the mutation in codon table, and that it is to their detriment if you're looking at functional prediction. So we're going to do a little experiment. Let's take a naive B cell sequence. We have its DNA sequence here. We're going to look at its protein sequence and do the masked language modeling thing. So we're going to do masked language modeling at each site. So let's take this site, for instance. We'll look at the original codon. That codon codes for a Q. So we're going to do the mass language modeling thing, but all we're going to do is stratify the predictions into those that are accessible, those amino acids that are accessible via a single nucleotide codon mutation. Those are going to be in the blue bin. And we're going to separate them from those that require two or more nucleotide mutations to change. So H and R are going to be in the blue bin, and W and N are going to be in the green bin. So we'll just do our masked language modeling prediction and then separate into green and blue. So the slide I'm going to show next is going to have 1 point for each amino acid at each site. And for each combination, we can evaluate, is it a single nucleotide away from the wild type codon, or is it multiple nucleotides away from that wild type codon? And the effect is not subtle. The amino acids that are accessible via a single mutation in a codon are judged to be 100 times more likely almost than those that require a multiple mutations in a codon. That is a striking effect. And when you think about it, it's not that surprising. Because when you think about how is ABLang trained, it's trained on seeing observed sequences. And most of the time, the majority of the time, those amino acids, those codons have 0 mutations in them from the parent sequence. But sometimes they have 1, and rarely they have 2 or 3. And that's exhibited just right here. So that means that ABLang is judging these amino acids that require multiple mutations to be 100 times less likely than those that require 1. But here's the catch. It's not that surprising that this is happening, but it's not good for functional prediction, as we'll be seeing later. Here's another thing. There's somatic hypermutation rate. As the people in this audience well know, somatic hypermutation varies a lot base by base, and it's influenced by the local sequence context and so on. So let's see, is ABLang2 influenced by somatic hypermutation rate? We're going to make a scatter plot in the next slide, and that scatter plot is going to have on this axis, what is the probability of non-synonymous change under a mutation-only model that removes the effect of selection? So if people are familiar with the S5F model, it would be something like the S5F model. It's not quite, and I'll tell you a little bit more about what it is later, but it's something like that. So this is a model of neutral mutation. On this axis is going to be the probability of non-synonymous change according to ABLang2. So what we mean in that context is that if we mask out a given site and we look at what is the probability of all the other residues according to ABLang2, how much probability mass is there? So how much does ABLang2 think that this thing is going to mutate? And if we do this for 9 sequences, randomly selected from a repertoire, we can see a really strong effect. Again, between this neutral probability, so the probability of mutation under a mutation only a model, not incorporating selection, and then the ABLang2 log probability of a non-synonymous mutation. I have brought these triangles represent points that I have brought into the Y range, but still the effect is very, very clear. If you do some sort of statistical test, each one of these has a correlation that is below machine precision in terms of p-value. So juxtaposing these two things, I think we can conclude that nucleotide level processes are important determinants of language model predictions. And next, I'm going to show that this has a negative effect on their ability to predict functional properties. So I will show that using this lab experiment. So we're not in computational world anymore. This is a lab experiment that came out in 2022, and this is called a deep mutational scan of these antibody sequences. So in a deep mutational scan, they mutate every site to every alternative amino acid and see what happens to the phenotype. In this case, the 2 phenotypes that were being measured, the 2 functional properties, is 1 expression and also binding to the target. And so here, for instance, if it's blue, that means it improved expression. And if it's red, that means it was bad for expression. So the task that we are going to set forth for Abilene 2 is to predict these deep mutational scans. You put in the parent sequence and then see, can it predict whether there's like the different amino acid site combinations are good or bad for expression. So we'll do a correlation. And FYI, I have flipped the perplexity here. This is the same type of correlation we looked at before, but I just like the y equals x, so I've flipped the perplexity. So up here are the ones that Abilene-2 thinks are compelling antibody sequences, and down here are the ones that it thinks are not. This is expression on this axis. So here we can see quite a reasonable correlation between expression and perplexity. So the model is doing a nice job of deciding which of these mutations are good or bad for expression. Here's the kicker. When we look at amino acids that are multiple mutations away from the naive, the correlation drops from almost 0.5 to about 0.3. And you can see also that the perplexity is evaluated as being substantially lower for these multiple mutations. And that corresponds to the fact that the model thinks that the amino acids that are farther away are inherently less likely. But we can see that this is detrimental to its ability to predict, because the codon table does not predict function. It predicts something, the effect of mutation. So to summarize the first part of this talk, we know that affinity maturation, which is a type of evolution, is the product of a mutation process, which is independent of functional properties, and a natural selection process where functional properties determine the outcome. Masked language models are modeling the entire thing, the combination of these mutation and selection processes. And we have seen that this combination is bad for predicting a function. So what to do? I would argue that Darwin has been staring us in the face the whole time. So we have a process of mutation and selection. Why don't we just model it with two separate terms? One, a mutation term that has to do with the probability of mutation given the parent nucleotide sequence, and also a selection term that has to do with the amino acid sequence, which I've denoted by P bar here. So people have been fitting, in the evolutionary biology community, people have been fitting these types of models, which were started by Halpern and Bruno. They've been fitting these things for decades, but they've been relatively simple. Here we have a lot of data, so there's a really exciting opportunity. We can fit richer models. So these are going to get an upgrade. So the P term, instead of being a simple model like an HKY, is going to be a small convolutional neural network. And I'll mention a couple words like this, but it's just going to be a small neural network to denote the process of selection-independent mutation. The F term is going to be what we call a deep amino acid selection model, which is a big model of the same flavor as they have for these language models for antibodies. It's going to be a transformer. And I'll also call it a DASM for a deep amino acid selection model. So if you don't know what a transformer is, it's okay. I have a slide, which is this. So hopefully this explains what a transformer is. Actually, there's many great tutorials about transformers, including the 3 blue, 1 brown one on YouTube, which I really recommend. So just briefly about the mutation model. And I'm not really going to go into it. We have a paper which is up on eLife. It's under revision, but you can imagine it like S5F again, if you know about that from the Kleinstein group. But instead of the model being a 5-mer, it has a wider window, a wider context window. And so what we've done is we've used embeddings. We use 3-mer embeddings in a way that we can increase the context window without the parameter penalty of increasing it. So typically, if you go from 3-mer to 5-mer to 7-mer and so on, you have to estimate an exponentially growing number of parameters. With this embedding, you get to increase the context window with only a linear penalty in the number of parameters. So we can estimate a 13-mer model for the same sort of parameter price as a 5-mer model. But for the purpose of this talk, the only important things is we have a model for how somatic hypermutation works that's estimated with just out-of-frame data. That's a separate data set than what we'll be using to estimate this natural selection model, which will be only functional sequences. How will we fit this selection model? We're going to use a strategy that I think we sort of borrowed from the Spisak et al. Paper from the French group, Alex and Thierry. So we take a bunch of repertoire sequences. Let's say this is each one of these is one individual. We can take all those repertoire sequences, we can group them into clonal families. So clonal families are collections of BCRs that emerge from a single VDJ recombination event. We can build trees on these because they are all clonally related. We build a tree with a simple phylogenetic model, and we reconstruct the ancestral sequences. So we call those parent and child sequence pairs. For each edge, we have a given parent-child sequence pair. So we want to model the development of the child sequence as a mutation and selection process. And our objective is to predict the child nucleotide sequence. And we do so using maximum likelihood. So we are going to tune the parameters of our natural selection, this DASM, this selection model, in order to maximize the likelihood of observing the child nucleotide sequence. So just to make sure this is clear, imagine we have a certain mutation that seems likely in terms of just the mutation process, but it doesn't appear much. We're going to say that that corresponding amino acid substitution was probably negatively selected. And we'll see lots more examples of this. But the idea is we're doing maximum likelihood fitting of the child sequences given the parent sequence. We also incorporate a notion of evolutionary time. So what do we get after we train such a model? Well, we get a model in which we can take an arbitrary amino acid antibody sequence, we can put it through our model, and we get out a table that's sort of analogous to this deep mutational scan, but this is selection factors. These are now model estimates. So now blue means that the model estimates this amino acid mutation, meaning mutating this site to this amino acid, is going to be beneficial in the course of affinity maturation. We are more likely to see that mutation than we would expect under a neutral mutation process. On the other hand, this one here that's red says don't mutate site 22 to a G because the model estimates this amino acid mutation to be deleterious. I want to emphasize that there's no functional information here explicitly given to the model. We are not giving anything, any stability measurements, any expression measurements, anything like that. It's just repertoire data. And we're trying to predict these child sequences, and we're fitting this selection model. It's also a mishmash, a collection of lots of different antigens. We're looking at repertoire data. This is not sorted repertoire data. It's just whatever that person happened to be exposed to is what we are fitting. So in a sense, we are fitting antigen agnostic models of affinity maturation here. So we'll start by saying how well do these models predict the child sequences? We're going to do an experiment where we'll take a held out data set and do the same thing. We have these parent and child sequences, but we have what we call observations, which is just where are the non-synonymous mutations going from parent to child. So of course, when I say observed, I'm putting it in quotes because we were not there watching this happen, but we call it it's like a computational observation where we did our ancestral reconstruction and we said, okay, we observed in the data that there was a non-synonymous mutation here. We're going to compare the frequency of those observations, their counts, to model predictions. So the model predictions are where you put in the ancestral sequence, you evaluate what the probabilities are of non-synonymous mutations, and you sum together lots of probabilities, and you get an expected number of observed mutations under the model. So we're going to be carrying the actual observations to these expectations. The observations are going to be given dots, and the model predictions are going to be given bars, and we're hoping that the bars line up with the dots. So we're going to predict the child in like 20,000 parent-child pairs in a held-out data set, and we're going to align these according to the IMGT numbering scheme and aggregate the results. But I want to emphasize that all this prediction was happening on an individual sequence basis, and then we align it according to IMGT and then aggregate. So here's what it looks like for ABLANG2 if we mask out sites and ask it to predict where the amino acid substitutions are. The bars don't line up super well with the dots. And I think that this probably has something to do with the fact that it always probably wants to mutate things back to the germline. But it is not especially well suited to this task. Whereas if we put together our mutation model in DASM, it's doing a rather accurate job of predicting, which is predicting where are there going to be mutations in affinity maturation. These are non-synonymous amino acid mutations. So just to recap again, so this says that for IMGT site 64, the observations, the number of observed substitutions was this, and then this is the prediction. There's sort of some little hatching in there for error bars. Okay, so these models do a good job of figuring out where in the sequence you have these amino acid substitutions. What about what amino acids they do predict? Do they do a good job of predicting what amino acids are there? So for that, we're going to calculate something I call the conditional perplexity. So the conditional perplexity is where we say, let's assume that we know that there's an amino acid substitution at this site. And we're going to say, calculate a perplexity of all the other amino acids that could be at that site. We're going to get probabilities for all those other things. We're going to condition on there being an amino acid substitution. So how well do we do predicting among all the other possible amino acids other than the parent amino acid? How well do we do in predicting that alternate amino acid? So here we're looking at two distributions of this conditional perplexity. And so ABLANG is in this sort of mustard color. The mutation model plus DASM is in purple. And we can see that the distribution is shifted to the left for the DASM model, meaning it's doing a better job of predicting the alternate amino acids than ABLANG2. Again, I don't mean to be picking on ABLANG2. I think this is going to be representative of all the language models. We also see that the DASM has significantly fewer outliers. And if we're looking at the medians, let's try to interpret these. So a median of 5 would mean that we have a 1 in 5 chance of predicting the correct amino acid. We're doing a little bit better than 1 in 5. For ABLANG2, it's a little bit worse than 1 in 7. So we would never pretend to imagine that that we would do a perfect job of this. That's sort of abiological. There is inherent stochasticity in what substitutions happen, and that there are lots of amino acids that are suitable at a given site. I wouldn't say lots, maybe on the order of 3. But there is a number of different amino acids that work at a given site of an antibody most of the time. So I mean, I would never expect this number to get less than 3. I'm sure we can do better, but just to ground that the fact that it's 4 isn't causing us to tear out our hair. Okay, so now let's move to functional prediction. So we're doing the same plot before. So we saw exactly this plot earlier. So this is expression versus ABLANG. Again, this is flipped perplexity. So we want to see a correlation on the x equals y. And we can see right out of the gate that the DASM is predicting the expression significantly better than ABLANG2. This is for the amino acids that are a single mutation away from the wild type. I just wanted to really nail down that like this expression, this is a lab measurement and this is a model prediction. But I think really the exciting part happens when we look at the things that are multiple mutations away. So here we maintain our correlation. It still looks good in a situation where masked language modeling gets confused by the codon table. So putting this together, we see a correlation that looks significantly more compelling, at least to my eyes, than this correlation over here. So I could show you many, many, many such plots, but rather than that, I'm just going to summarize them into a table. So what we saw just now was the comparison of ABLing2 with this expression data set to the DASM, which got almost 0.67. So we can also look at other models. So ESM2 is the Facebook model. It's trained on lots of proteins. It's like 650 million parameters. ProGen2 was a really nice effort by Salesforce research back in the day when companies thought it was cool to do these things. And it's second best to DASM. It does quite a reasonable job of predicting expression. And that's interesting and cool. So there's other data sets that we can evaluate. We can look at the binding component of that Koenig data set. And our correlation drops. We're still substantially better than the second best. These two are binding data sets. They're really one data set where we've stratified into the different lengths of CDR3. And somehow ProGen2 can't predict binding at all here. ABLing does a somewhat reasonable job, but we're doing a substantially better job for those. I wanted to say that ProGen2 in that Flab paper was viewed as being, this is ProGen2 small, it was viewed as being the best performer overall in terms of these different things. So we're not finding some crappy model to compare to. So another advantage is that for these masked language modeling things or an autoregressive model like ProGen2, you have to do this sequential masking thing in order to evaluate probabilities of alternate sequences. So it takes about 8 seconds per sequence for ABLing2 because you have to do the sequential for a high quality prediction, you have to do the sequential masking. And ESM2 is substantially more, ProGen2 is quite a lot. But ours only requires 0.04 seconds per sequence, and this is averaged over evaluating 100 sequences on an A100 GPU. So the reason, again, is like we don't have to do any sequential masking. We just put in the full parent sequence, and then you get a full prediction. Our model also has about 1/10 of the parameters of the smallest competing model. So that's all fine. I mean, maybe I've beat my chest for the machine learning crowd. I'm sort of more interested, can we learn something interesting biological from these models? So in order to do that, I will just remind this audience who probably knows a lot more about this than me about the structure of antibodies. So they have a heavy and a light chain, and they have 3 fingers that reach out and touch the antigen, the CDR1, CDR2, CDR3. So what we can do is we can evaluate these models on lots of sequences and then look at the aggregated selection factors per site. So I'm shifting gears a little bit. Before we were looking at a per site per amino acid level. Now we're going to look at a slight model variant where instead of outputting 20 dimensions, we're just going to output 1 dimension. We call this a deep natural selection model. And that 1 dimension for each site of the sequence is going to indicate, are we looking at diversifying selection or purifying selection? So purifying selection means that modifying that amino acid happens more rarely than you would expect under neutral mutation. And diversifying means that it happens more often than you would expect under neutral mutation. And so what that intuitively means is that you don't want to modify the things that are under purifying selection, and you do want to modify the things that are under diversifying selection if you want to have a nice story of affinity maturation. So we're going to align those according to the IMGT numbering and aggregate on a held out data set lots of different model predictions. So we are also going to stratify the sequences in terms of do they use IGH v1, v3, and v4. So just to put it all together, so this point here is the median selection factor for all of the IGH v1 sequences at IMGT site 14. So this is predicting that they're evolving roughly neutrally, which is kind of interesting and cool. Down here are the ones that the model is predicting should not change under affinity maturation. And when there's a highly conserved site, I put down the amino acid that's down there. So here is our big plot of the heavy chain selection factors. I've explained what all the elements mean, but I just also want to explain that this is wrapping. So we go to site 65 and then we wrap around and we get to site 68. So let's zoom in a little bit here and see what we can see. So for people who are familiar with antibodies, this should not be surprising at all. The model estimates that there's strong purifying selection for the cysteine and tryptophan residues that bound the CDR1 and those that bind the CDR3. So this cysteine tryptophan have among the lowest selection factors. But there's some other interesting stuff here. Like what is this R? And what is this RDY? Those are also estimated to be under strong purifying selection. And that wasn't something I had ever heard about. Oh, heard about before. Well, if we do a structural analysis, and by that I mean if I ask Phil Bradley to look at this structurally, he looked and said, well, this RRDY is sitting at the base of the variable region and it's making a lot of bonds that are presumably stabilizing the base of the variable region. So that's cool. So we're already seeing at least things that I don't know. So it does turn out that this has been reported in the antibody structure literature. It's called the charge cluster. But when I talked to lots of antibody structural biologists, they didn't know this offhand. But this is an important thing according to the model. Let's take a closer look at the CDR3. So the CDR3, you would think it's under really strong diversifying selection, and it sort of seems very variable. And so it seems worth looking at in a little bit more detail. So we are going to stratify by CDR3 length. There's this, it's annoying to compare the CDR3 until you stratify by CDR3 length, and then you can look at each site individually. So let's do that. So this is a motif plot of just the presence of amino acids in the ancestral sequence, meaning the parent sequence. And then over here are the selection factors. So I want to draw your attention to this second to last site that's frequently a D. And we can see that this site is frequently under strong purifying selection. Again, this is stratified by CDR3 length, and this is also stratified by CDR3 length, so we can make suitable box plots. So this is a different view of that same thing, but it's a summary. So along here is CDR3 length, and then on this axis is the selection factor. Each point represents a single site of the CDR3. The first site is this yellow one, and this last site is this purple one. And the second to last site, which is mostly purple, is the one that I noticed was under strong purifying selection consistently. When it's a D. When it's not a D, it's nothing special. It has similar selection factors as the others. So I think I just want to set up that I think this is interesting. People usually think that the CDR3 is this thing that is meant to be mutated. You should just change it however you want. But here we can see that there's really strong purifying selection on this D. And there's no way we would have been able to do this without some sort of more complex model than just looking at the V genes per site and calculating selection factors. So again, we can look structurally. And Tyler Starr, who's the collaborator, when he looked at this, he saw, oh, look, there's a salt bridge that happens at the base of the CDR3. And Phil looked at some other structures and he saw, well, sometimes this D is making connection with the light chain and also with the backbone on the other side of the CDR3. So this is just another sort of vignette of how we can use these models to learn something interesting about the way antibodies work. And to end with a mystery, you can see that sometimes there is a lot of diversifying selection outside of the CDR3s. And this is or outside of the CDRs. And this is kind of weird. And Gabriel Victoria said, I don't really believe this because if there was diversifying selection happening outside of the CDR3s, why wouldn't it have happened in the evolution of the germline genes? And I don't have a really good answer to that, but let's just look at an example of this. So this is just a little view. This is an example antibody. And this example antibody, we'll just look at these two sites that are not in the CDR3 that are under diversifying selection. This one and this one. This is software from Jesse Bloom's lab. It's really neat. It's called DMS Viz. So what are these two sites? Well, this one is just before the CDR3. It's a serine that often mutates, a threonine that often mutates a serine. And down here is a proline. And like, if I was to pick sites that I thought were under diversifying selection for this particular antibody, I would not pick these sites. This one is sort of sitting on the outside, and this one is very deep inside. But, and I'm happy to go through this if people want. We can do an independent counting argument and show that this is indeed the case. It's not just a fault of the model. Okay, well, I'm going to wrap up here. Evolution is a product of mutation and selection, and we win by modeling these two components separately. Moving forward, we're really excited to extend this to other settings. So if we think about the evolution of viruses, for instance, in the evolution of viruses, one often wants to estimate natural selection by taking a single collection of sequences, aligning them, and making a tree. And if you're lucky, you have a lot of data and you can estimate a per site selection model. But here, the idea is, what if we take lots of different, not explicitly related evolutionary histories, but similar evolutionary histories? We train as given DASM. So these could be, for instance, hemagglutinin in different like H1N1 versus H3N2 and so on and so forth. And then we get a model that gives you a model of maximum resolution. So then we can put in an arbitrary sequence. And so for that single sequence, we can get out a per site, per amino acid prediction. And we'd like to actually take this to ESM scale where we train it on the known protein universe. And so to conclude, I think mass language modeling has shown incredible success for human language. I mean, GPT is, I think, one of the most important things to happen in recent technological development. But that doesn't mean that using exactly those same sorts of frameworks is optimal for biology. And I hope biologists get involved and think about what these models are doing and how we can improve them. So I'll stop there. I'm grateful to many people. I encourage people to email me. It's my favorite form of social media. If you don't have an email account, I encourage you to get one. Um, and I would also like to thank all the people that have contributed to this work. This is our core team at Fred Hutch. Yeah, Mackenzie and Kevin were really important in doing the data preparation and thinking about how all these models work. Will and I have been coding side by side on these models recently. Hughes helped sort of advise and guide things along with Julia. These are collaborators that have just helped advise. I'm more grateful for funding than ever. Thank you, Naiad and Hughes. Thank you so much to the AIR community for being amazing through all the years, and I'm hoping to re-engage more moving forward. Of course, antibody sequencing labs, we've had some great help from Corey. Database curators, Michael Chagyan, who led the FLAB project, which made it so much easier to do these sorts of comparisons. And definitely these AI models for helping us write the code. And I'll stop there.
Thanks, Eric. This was really exciting talk. I just looking if there are questions from the audience. If not, I will start with my questions. So when you showed, so starting with a very simple question, when you had in the middle of your talk this table where you showed the performance across the different language models, did you try and when you tried and to predict binding and this one I think this one. Did you try also? So I know here the question was just to compare language models, but what we have seen in our research or when we have looked at it, that many times we don't see the advantage of language models over very simple approaches like random forest or something like that? Did you benchmark against very simple models to see if language models are better in general,
So
or
what I think you're talking, so this is, I should be clear, this is zero shot prediction. There's no labeled training data.
oh, okay. Oh, okay.
So I don't think that there's a way you can use
No, you
a
cannot.
random forest to make a zero shot prediction. So there's no fine tuning. There's like Kleinstein and others have done interesting things where you take the protein language model embedding and then do a little bit more machine learning to train a classifier for a specific prediction task.
Yeah.
This is totally zero shot.
Okay.
These models have never seen this data and they're asked just from scratch to predict the table.
Okay. Got it. Got
But
it.
great question. I mean, I think I would just say that like in general, like we need to be honest and clear about what good baselines are. So I appreciate the question very much.
Yeah. And so then the next question. So this is in vitro data that you're showing here, right? This is from deep mutational scanning, some kind of in vitro data. So can you just give me, maybe it's a stupid question, but can you give me an intuition of why learning affinity maturation rules or incorporating B cell evolution helps with predicting in vitro mutational data, which doesn't have to follow the rules of in vivo evolution.
Yeah, great question.
Um,
So if you were just imagining a B cell evolving, and remember, the goal of this B cell is to have a good binding antibody, and it encounters a destabilizing mutation in the antibody,
mhm.
that's going to get wiped out
Yes.
in the selection pressure. So we're not going to see that mutation as frequently as we would expect
Mhm.
under a neutral mutation process.
Yes.
So although affinity maturation is not an in vitro thing, I mean, it's both the in vitro and the germinal center are in a sense learning about functional constraints. I mean, still at the end of the day, there is a notion of antibody stability. Both these situations, you're interested in stable antibodies. And so this is exactly what the model is trying to do, is trying to understand, given the mutational processes that are at play in affinity maturation, how do we learn the selective constraints, which are entirely on the functional plane?
Mm. And then, so you saw that or you showed in this table that you have an improvement, but there's probably a ceiling. And is that ceiling because you're not um, incorporating the antigen in the model, that you're not incorporating the epitope? So that, yeah, probably yes.
Yeah, there's a couple things. I mean, so the natural affinity maturation process is different, like you say, than the lab environment. So there's more complicated constraints that happen in affinity maturation. The other is these assays are noisy. I mean, if you look at a deep mutational scan, even from highly skilled labs like Jesse Bloom's, they do not always get like perfect correlation. Sometimes they get really, really great correlation, but like I mean I don't like I'm not an expert in these things. I think that there is, but I mean when I talk to Jesse, he's like, well, there's a certain amount of noise going in these sorts of things. So there's that. Also, as you say, we don't like the antibody selection model does not know anything about the antigen, but here I wouldn't think that that would affect our expression.
No
Uh,
expression, not, but the binding predictions. I'm sorry,
yeah.
the binding
And I
prediction.
think it's actually sort of interesting and mysterious that the model is able to predict anything about binding
Yeah.
affinity at all. And I would ascribe that to two things. So there's a certain component of binding that is also just is it a stable antibody? So you can't have good binding if you have a really unstable antibody. So perhaps there's part of that. The other thing is, I think that there is implicit information, and we have some ideas about how to like refine this information. There's implicit information in the fact that you have lots of sequences in a single clonal family that are all evolving against a single antigen.
Mm.
And so it's possible that the model is recognizing, oh, I know this. And I mean, even though I don't explicitly know what it's binding, I recognize that when you see an antibody like this, it's going to evolve like so
Mm,
because it tends to evolve against a given antigen, if
mm.
that makes sense.
Yeah, yeah.
So,
Oh, and what you're also maybe meaning is that there is some kind of confounding, if one can say it, between expression or stability, as you say, and binding,
yeah,
maybe.
definitely.
Yeah, yeah. So we would need experiments that actually can differentiate, ideally between.
Yeah, I mean, so for instance, TightSeq is actually good at doing this from uh, Justin Kinney and Alex Thierry.
Yeah, yeah. It would be great to have more tight seq data than the few data sets that are out there. Yeah.
Yeah, it's coming like there's Timothy Whitehead is doing more of these sorts of experiments, and that's getting incorporated into the next version of Flab. So I'm looking forward to evaluating on that.
Mm, right. Yeah. Um, yes. I think these were my questions so far. Are there any other questions from the audience?
Great.
The shy audience there. Okay. Thanks, Eric. Thanks so
Yeah.
much.
Thank you, Victor.
Thank you.